Hello, one and all. This is Ayush Shetty from TYBSc Microbiology. My seat number is 2315184. I'll be presenting to you about the Western blotting technique. Western blotting is a method by which the proteins can be isolated and separated on the basis of their size, shape, molecular weight, and electrical charges they possess, and eventually by its antigenicity to a specific antibody. The Western blotting technique is an assay used to detect various viral proteins. Here are a few terminologies which surround around the topic Western blotting, such as separation, isolation, SDS page, electrophoresis, nitrocellulose membrane filter, antigen antibody reaction, chemiluminescence, radio labeled antibodies, viral proteins, and so on. This gives us an idea about how Western blotting technique is performed and what are the key components or steps of this bioassay. In Western blotting, a protein mixture is electrophoretically separated on SDS polyacrylamide gel. The protein bands are transferred to a nitrocellulose membrane post electrophoresis, and the individual proteins are identified by flooding the nitrocellulose membrane with radio-labeled or enzyme-linked polyclonal or monoclonal antibodies specific to the protein of interest. The antigen-antibody complexes that form on the band containing the protein are recognized by antibodies, which can be further visualized in a variety of ways. The assay is further used in the diagnosis and proceedings of various viral infections. The set of proteins received in a sample are first denatured by the means of SDS, which is sodium dodecyl sulfate, which is an anionic detergent and helps in breaking various non-essential bonds between the large protein molecules so that the antigenicity of the protein remains intact. This SDS treated protein mixture is then loaded onto the polyacrylamide gel so as to electrophorese it further. The gel is electrophoresed in the direction, and the direction of the migration is from the positive terminal to the negative terminal. From the negative terminal to the positive terminal, as the proteins, which are a polymer of amino acids, are anionic in nature. The gel post electrophoresis is removed, and then the electrotransferring of the electrophoresed protein components is done on a porous membrane sheet, which is mostly nitrocellulose membrane. The nitrocellulose membrane receives these proteins from the gel by means of application of electric field. This nitrocellulose membrane is then separated from the gel and flooded with the antigen of interest, which is either an enzyme linked or radio labeled antibody. Upon incubation, for a certain period of time, the excess of antibodies are washed away, and then the screening is done by means of either chemiluminescence or radiography. Enzymes specific to the desired proteins may be fluorescent or radio labeled as well. The step Y comprises of visualization of the same, which can be done either by radiography for radio labeled antibodies, while chromogenic substrates are added so as to amplify the results of chemiluminescence. The various applications of Western blot technique can be seen in the studies of protein protein interaction, protein isoform detection, antibody characterization, and epitope mapping in terms of medical microbiology, subcellular localization of proteins, genetic studies such as post translational modification, protein DNA interactions, etc. Thank you. The citations of the references are listed over here. Thank you.